Welcome to lecture 13 on fatty acids uptake, transport and storage and catabolism. You will find a source for this lecture in Leninger chapter 17. Focus on the pages that I've indicated here. Before we start, I want you to remember the numbering and the nomenclature for fatty acids. Here, focusing on this particular fatty acid, the carboxylic end and the methyl end. In particular, this carbon, the beta carbon here, which is the second after the carboxyl group, so carbon number 3, but it is also called a beta carbon. It will be important to understand how beta oxidation occurs. And this slide is to remind you about the structure of a triacyl glycerol, which is 3 fatty acids here esterified onto a glycerol backbone. Cells in our body receive fatty acids as a source of fuel and energy from different sources. One of these sources is from the diet. And following a meal, fats and other molecules are ingested and end up eventually in the small intestine. And they are emulsified with bile salts. These bile salts are derivatives of cholesterol, and they allow a mix of fatty acids and triacyl glycerol that are available for digestion by lipase, these intestinal lipases. So these will degrade these triacyl glycerol molecules into fatty acids. And these fatty acids will then be taken up by cells that are lining this small intestine that are called enterocytes. These are epithelial cells, and these will take up fatty acids, will repackage these fatty acids into a tag molecule. These molecules will then be gathered into assembled into a particle called a lipoprotein, and a particular lipoprotein called chylomicrons are specifically generated by these intestinal cells to allow the transport of these very hydrophobic molecule inside a particle. So let's have a look at a little bit more in detail at this particle. It's made of a monolayer. Here you can see this monolayer of phospholipid, of glycerophospholipid, sometimes intercalated with cholesterol, so free cholesterol. And inside, in the core of this particle, you will find all of our triacylglycerides molecule packed in there. In addition, in the core, you will find some esterified cholesterol, which is fully hydrophobic, because the hydroxyl group here, the only polar group in the whole molecule, is esterified with a fatty acid. So these cholesterol ester are also found in the core of this particle. In addition, on the surface of this monolayer lies some proteins that are called apolipoproteins. They're of different nature, and these can be recognized by some receptors on cells where these particles are traveling. So these particles, these chylomicrons, will travel in the blood and will allow transport of our fatty acids in the form of this esterified molecule. Now we come back to this slide, and we have our chylomicrons that travel in the blood circulation. They end up in some small blood vessels, capillaries, that are in the vicinity of some tissues, for example, muscles or adipocytes. And on the surface of these cells, facing the lumen of the capillary lies these lipoprotein lipases, which will be activated and will convert our triacylglycerol from the chylomicrons to fatty acids. These fatty acids are then taken up by either myocytes or adipocytes, and these are then repackaged in the cells in a tag for storage of energy in adipocytes, or these fatty acids can be used and oxidized and used as fuel through this beta oxidation that will go into more details. This picture represents a section of a white adipose tissue. It consists of many cells that are packed with tags in these white translucent areas. So there's a huge lipid droplet in these cells, and this is the job. The cells are committed to store fatty acids esterified in tags in these lipid droplets here. Here is a representation of an adipocyte here with its lipid droplet that I mentioned earlier on the previous slide. This lipid droplet is also lined with a monolayer of glycerophospholipids, and the tags are inside in the core of this lipid droplet. Now, these fatty acids can be used as fuel source, as an energy source, and needs to be released and mobilized from this tag. And this happens in the case of low levels of glucose, which will lead to the secretion of glucagon, which is a hormone with opposite action from insulin. So glucagon will then act as a ligand, activate the receptor on this adipocyte, which is a G-coupled protein receptor, which will lead to the activation of a G protein and the activation of adenylyl cyclase. This enzyme produces cyclic AMP from ATP, and cyclic AMP acts as a second messenger. It's a second messenger which activates a protein kinase A. Protein kinase A has many substrates, as many of you have covered in their assignment, and some of these substrates are lipases, which allows the mobilization of fatty acids from triacylglycerols. What does it do exactly? Well, it phosphorylates perilipin, which is a protein that lies on the surface of the lipid droplet. Once it's phosphorylated, it dissociates from this cofactor, CGI58, which will then be available for allowing of the mobilization of this lipase, adipose triacylglycerol lipase. And this particular lipase will act on a tag and release one fatty acid and diacylglycerol here. Then the phosphorylated perilipin will then associate with hormone-sensitive lipase, which is itself also a substrate of pKa, which allows its binding to perilipin and its association with the lipid droplet. So this lipase can then find its substrate, which is diacylglycerol, will liberate one fatty acid, and then will end up with the monoacylglycerol. Finally, this monoacylglycerol can then be catalyzed by a monoacylglycerol lipase, which will release the last fatty acid and glycerol. So these fatty acids can then be transported, can be exported out of the adipocyte and then transported in the blood circulation in association with serum albumin. So this is a protein that binds fatty acids and then shields their hydrophobic acyl chains and allow their transport into an aqueous environment. until they are transported into, for example, a myocyte. This is a muscle cell, which will then use this fatty acid as an energy source, and via its beta oxidation, so generating eventually ATP. Here is a summary of the fate of this tag molecule, which is mobilized to release fatty acids at each step. So it's a summary which might be easier for you to see the action of each of these lipase in this slide. Here it's indicated, fatty acids are indicated as non-esterified fatty acids because they are not esterified onto the tag molecule with the glycerol. In summary, what happens to our tag molecule when it's ingested and reaches the intestine? It is subjected to lipase catalysis into fatty acids. Then these fatty acids are repackaged in the intestinal epithelial enterocytes into tags. These are transported out via a lipoprotein called chylomicrons, and a new set of lipase called lipoprotein lipase will release fatty acids from this tag. These can be taken up by adipocytes for their storage as tag, or under low glucose conditions, these can be mobilized and fatty acids can be released via the successive action of lipase that we've just looked at in the adipocytes. Release of fatty acids, these can then undergo beta oxidation in the muscles, for example, or in tissues that require energy. So far, we have focused on fatty acids and how they are released from tag, but we also obtain a molecule of glycerol, which can also enter glycolysis, and this is how it happens. So glycerol is first phosphorylated by glycerol kinase, which is then reduced to dihydroxyacetone phosphate, which can be isomerized to the glyceraldehyde 3-phosphate, which is an intermediate of glucose, of the first glucose steps of glycolysis, and then this enter glycolysis. So now let's use our fatty acids. And in contrast to the biosynthesis reaction that we've covered to generate and produce fatty acids, in the case of catabolism of fatty acids, i.e. beta oxidation, this occurs in mitochondria. So these fatty acids need to be transported from the cytosol into the mitochondria, and this is done via an acyl carnitine and carnitine transporter. This takes place with the 3 reactions that we will focus in the next slides. Before a fatty acid is transported inside the mitochondria, it is first activated in the cytosol by fatty acid CoA synthetase. Here it is, our fatty acid represented with an R, which is any hydrocarbon chain here, and it is conjugated to coenzyme A. So this is the first step that will look into more details. So here is the reaction summarized here, 1 molecule of fatty acid plus coenzyme A here with its reactive thiol group and 1 ATP, which will lead to fatty acid coenzyme A, AMP, and pyrophosphate. Here is the detail of the reaction. Here's our fatty acid, and this carboxylic group will be adenylated here to form a fatty acid adenylate plus pyrophosphate. This will then be hydrolyzed to 2 inorganic phosphate, which will generate energy and drive the reaction further via the thiol group of our coenzyme A, which will attack the acyl adenylate here, displacing AMP. and therefore generating a fatty acid CoA. Let's pause for a second and let's look at our fatty acid CoA. If you remember, this is also a molecule that was used during biosynthesis of TAG, of the first step generating phosphatidic acid and then generating TAG. But this activated fatty acid under conditions of low blood glucose and when the system needs energy, so this fatty acid, this activated fatty acid can go inside the mitochondrium for beta oxidation. So this molecule can therefore be used in the cytosol for anabolic reactions or catabolic reactions in the mitochondrium. So now that we have our activated fatty acid, it will be trans-esterified onto a molecule called carnitine on this hydroxyl group here that will allow its recognition on the outer mitochondrial membrane. This conjugate can then diffuse in the intermembrane space and can be recognized by this transporter, which will allow the transport of this of our fatty acid carnitine inside the matrix of the mitochondrium. Then this will allow the release of our fatty acid, which is conjugated again with coenzyme A, and the release of carnitine, which can be then recycled to the outer membrane of the mitochondrial. We now focus on the catalytic reactions of our fatty acids, and we will first focus on a fully saturated fatty acid of 16 carbon, which is palmitic acid, here depicted here. And the first stage, stage 1, is the beta oxidation, i.e. it's the release of units of 2 carbons successively from this molecule. These will generate, each unit will generate 1 acetyl-CoA. So in total, for 16 carbon, we will generate 8 acetyl-CoA upon beta oxidation of palmitic acid. And then in stage 2, these acetyl-CoA will enter the citric acid cycle and generate NADH and FADH2. But this was covered in chapter 16 already, so go back to it when you looked at the fate of glucose. And finally, stage 3 will generate ATP from NADH and FADH2 via the respiratory train in the mitochondria. So let's focus on this stage 1 and the successive oxidative removal of 2 carbons from the carboxylic end of this 16 carbon fatty acid. It's depicted here, and it's activated, so in the matrix it's conjugated to coenzyme A, and we have our alpha carbon here and our beta carbon, and the rest is not depicted, but we have a total of 16 carbon. So each release of these 2 carbons is dependent upon 4 enzymatic steps that we will cover here little by little. So first of all, let's cover the first two steps. Now the first steps consist of an oxidation reaction, i.e. the dehydrogenation of an alkane to an alkene, so with a double bond here between the alpha and the beta carbon. And this is dependent on FAD, and these 2 hydrogens will be transferred onto FAD to generate FADH2. Then the second step is a hydration with 1 molecule of water, and this yields an alcohol here on this beta carbon. This is done by the NOL-CoA hydratase. So this is how FAD will accept 2 hydrogen and 2 electron onto the flavin rings here of this whole FAD molecule. So third step is an oxidation step, which undergoes dehydrogenation of this alcohol group into a beta keto-acyl CoA. Finally, transfer of a coenzyme A molecule onto this 14 carbon hydroxyl chain will release 1 acetyl CoA. The release of 1 acetyl CoA leaves 1 hydrocarbon chain of 14 carbon, so myristyl coenzyme A, which will go back to step 1 and undergo the 4 successive enzymatic reactions to release a new acetyl CoA, and so on and so on and so on. If we take a closer look of the 2 oxidation reactions during these 4 steps, there is a generation of FADH2 and NADH and H+. And electrons are then they pass through the electron transferring flavoprotein and then through another flavoprotein, which leads to the respiratory chain. NADH enters the respiratory chain via the action of an NADH dehydrogenase. So these actions replenish the pool of NAD and FAD. The sequence of reaction occurring during beta oxidation, i.e. this oxidation generating FADH2, hydration, and then dehydrogenation of this hydroxyl group here, are also found in reactions in the citric acid cycle and of the oxidation of some amino acids, as you can see, generating also FADH2 and NADH. Hence, the regeneration of FAD or NAD will lead to the transfer of electrons to the electron transfer respiratory chain. In sum, for 1 palmitic acid, we end up with 8 molecules of acetyl CoA, 7 FADH2, and 7 NADH. So these acetyl CoA enters the citric acid cycle and are further oxidized into CO2. This will make more GDP, NADH, and FADH. These will go into the respiratory chain. So all in all, 1 palmitoyl CoA will generate 8 acetyl CoA. In addition, 7 FADH2 and 7 NADH. These will generate 10.5 plus 17.5 ATP form. So altogether 28 ATPs plus 16 molecules of water. So fatty acid catabolism generate not only energy but also water. Now, these acetyl CoA enter the citric acid, the citric acid cycle, and they will generate themselves NADH and FADH2, which will in total, from 8 acetyl CoA, this will generate 80 ATP plus 16 molecules of water. So if we addition the equation from the slide before to these slides, to the fate of acetyl CoA, All in all, one palmitoyl-CoA will generate 108 ATP plus 23 molecules of water. But we have to subtract 2 ATP from the activation of palmitoyl-CoA. So all in all, 106 ATP are generated from one fatty acid, from one 16-carbon saturated fatty acid. So far, we have focused on the beta oxidation of saturated fatty acids. Now let's focus on monounsaturated fatty acids, i.e. fatty acids that contain one double bond. So there's a slight difference, but let's take the example of this olehyl CoA, 18 carbon and one double bond here at between carbon 9 and carbon 10. So the first steps are the same, i.e. the release of 3 acetyl-CoA following the 4 steps that we've looked into earlier. But this stops here due to the encounter of this double bond. These naturally occurring fatty acids with unsaturations have this conformation in cis here. There's 2 hydrogen on the same side. However, if you remember, the first step of oxidation of a fatty acid includes the generations of one double bond, but this is created in trans. The NOL-CoA hydratase that will recognize this the substrate containing an unsaturation does not carry out the reaction for cis double bond, but only for trans double bond. So an additional reaction is necessary, which will isomerase this cis double bond into a trans double bond. That means that we can continue at step number 2 directly for this for the release of this first for the next 2 carbons here. This means that we have skipped the step number 1 of the 4 steps, but instead have used this reaction to isomerize the double bond. This also means that since that we have skipped the first steps, we have one less FADH2 made for a monounsaturated fatty acid. What about polyunsaturated fatty acids, i.e. fatty acids that contain more than one unsaturation? Here is the example of linoleal-CoA, which is an essential fatty acid for mammals. It has 2 cis double bonds here in carbon 9 and 12. So as before, this fatty acid will undergo 3 rounds of beta oxidation following each for the 4 steps, which release 3 acetyl-CoA. And now we end up with these 2 cis double bond. So the first one will be isomerized just like we did for the monounsaturated fatty acid to generate a trans double bond here. This will then will undergo an oxidation of this cis double bond, which will then release a fatty acid with only one trans double bond that the NOL-CoA isomerase can take care of, and will continue in the next steps of beta oxidation, just like we showed it was shown for a monounsaturated fatty acid. This enzyme is NADPH dependent and not so the transfer of hydrogen from NADPH onto the fatty acid. If we look closer in detail here, we'll see that again, just like for monounsaturated fatty acid, there will be one fewer FADH2 generated after this isomerization because we missed the first step. But one FADH2 is produced during the first step of the next cycle here, because we oxidize this this other cis double bond. This means that this will result in no further loss of FDH2 because this will undergo the regular 4 steps of beta oxidation. Most naturally occurring fatty acids have even-numbered carbon and can undergo the beta oxidation that we've covered so far, whether they are saturated or unsaturated. But many plants and marine organisms also synthesize odd-numbered fatty acids. These odd-numbered fatty acids will undergo beta oxidation until we reach the 5 last carbon, which will release one acetyl-CoA and one propionyl-CoA. This propionyl-CoA, a 3-carbon compound, will require further steps for the final oxidation. And these are the 3 steps. This propionyl-CoA will undergo a carboxylation, and this reaction is a similar reaction than for acetyl-CoA carboxylase, which generate malonyl-CoA. And then this is followed by an epimerization here, i.e. the transfer of this this CoA onto this last carbon, hence generating succinyl-CoA. This can then go into the citric acid cycle. The regulation of fatty acid synthesis and breakdown is highly coordinated. Now, if we focus on the right and the beta oxidation steps that we've covered so far, the rate limiting is actually the entry of fatty acid CoA inside the mitochondrion. As soon as this fatty acid is transferred onto carnitine, and this conjugates is then transferred inside the matrix, beta oxidation occurs, so these steps are committed. So how is this regulated? So this is negatively, this rate limiting step is negatively regulated by malonyl-CoA. And if you remember, malonyl-CoA is the first step leading to fatty acid synthesis. So malonyl-CoA is generated from the carboxylation of acetyl-CoA by this acyl-CoA carboxylase, ACC. So this enzyme is also regulated by phosphorylation and dephosphorylation. When the system has high glucose, high blood glucose, high carbohydrate load, this will lead to secretion of insulin, which eventually will induce a phosphatase, i.e. will dephosphorylate ACC, which when it's phosphorylated is in its inactive form. So the dephosphorylation will lead to its activation, leading to acetyl-CoA carboxylation to malonyl-CoA. This is the rate limiting reaction, and then this will lead to storage of this acetyl-CoA into fatty acids, and storage of energy in the form of fatty acids. So when these steps occurs, malonyl-CoA is a negative regulator of beta oxidation. Now, when low blood glucose is in the system, this will, in opposite, activate the secretion of glucagon, which, as we looked into earlier, will lead to the generation of cyclic AMP as a second messenger, activate protein kinase A, and this will then phosphorylate ACC in its inactive form. This means that there is low level of malonyl-CoA, hence allowing beta oxidation to occur. So using fatty acid as the major energy source under low blood glucose. Acetyl-CoA can have another fate under certain conditions, and this is because acetyl-CoA enters the citric acid cycle due to the existence of oxaloacetate. Now, in some cases, oxaloacetate is depleted, and we'll go through some examples later. And in this case, acetyl-CoA has another fate and is converted into ketone bodies. So these are actually compounds, soluble compounds that are generated. And there are three forms of these compounds: acetone, which is volatile, which you can smell, acetoacetate, and beta-hydroxybutyrate. Here, beta, this is alpha, and beta-hydroxybutyrate. So these are transported into the blood to tissues. And in particular, they can give energy that is required for the heart and the muscle, but also the brain can adapt to the use of these compounds instead of glucose. Now, what are the conditions in which the citric acid cycle is not functioning and acetyl-CoA can't enter the cycle? So when oxaloacetate is low, so under normal conditions, this is all fine. Acetyl-CoA goes into the citric acid cycle, but under conditions that promote gluconeogenesis, and i.e. untreated diabetes, it's because this is under diabetes, there's low insulin. This means that glucose is not taken up by tissues, and glucose therefore can't undergo glycolysis and generate acetyl-CoA that goes into the citric cycle and generate oxaloacetate. Another condition is a severe starvation, so reduced food intake. These conditions will slow down the citric acid cycle and will draw off oxaloacetate into gluconeogenesis, because these cells that can't take up glucose, even though glucose is plentiful in the blood, but because of the lack of insulin in the case of diabetes, insulin leads to the uptake of glucose. So in the case of diabetes, this is not the case. So these cells need to generate glucose, and this is drawn by the use of oxaloacetate into gluconeogenesis. So these are the conditions in which ketone bodies are actually formed. So the acetyl-CoA, which comes from beta oxidation of fatty acids, will then generate these compounds instead of entering the citric acid cycle. So the first step, starting from acetyl-CoA, will join two acetyl-CoA together via a thiolase. This is the reverse reaction of the last step of beta oxidation, when there's a release of acetyl-CoA. So these two are joins, and the third acetyl-CoA is incorporated, generating a beta-hydroxy beta-methyl-glutaryl-CoA. HMG-CoA. So this is HMG-CoA synthase. Then 2-acetyl-CoA are then freed under following these reactions. Then, in order to traffic to other tissues, the coenzyme A must be removed. So acetone and acetoacetate and beta-hydroxybutyrate can then travel through the blood. So acetyl-CoA is then removed from this compound, which leads to acetoacetate. So acetoacetate can then be decarboxylated and generating acetone, or it can be used to generate beta-hydroxybutyrate. So this is volatile, and this will be transported. These two compounds can be transported in the blood to be used as an energy source. Now, once in the blood, this beta-hydroxybutyrate can then enter other tissues, including the brain, and can then be further transformed under successive reactions into acetyl-CoA. Now, acetyl-CoA in tissues that are not insulin dependent will be able to be used as energy production.